All right, people will start coming in. Again, I think I'm going to try moving.
Is it the router?
Hello, everybody.
Hello.
Oh, Suzanne keeps freezing up, just so everybody knows. We're trying to get that straightened out before we get all the way started. She's having some internet troubles.
Figured that.
Thank you. That's also kind of perfect, though, because I'm still not home yet.
Perfect.
So how's everybody's Monday been going?
It's been a Monday for me, I'll say that. How about everybody else?
Pretty good. Just making hash today.
Nice.
Ooh, what kind of hash?
Some bubble. I'll press into rosin.
Ooh,
Some fresh frozen.
awesome. What strain?
Washed some chili verde today. It did really well.
Oh my gosh.
You are.
I'm back for the 20th time. I'm having serious internet problems tonight, guys. Sorry. Can you guys hear me? Am I here?
Yep, you're here.
You are here.
Yeah, and they were out today. The cable company ran new lines to our house, and now it's worse. So, let me see. Is everybody, Katie, did you let more people in or?
No, I didn't.
Oh, no, I don't know if you can or not. I'm just trying to see if we're missing everybody. Oh, hold on a second. Here they are. Admit all. Okay, admit all. Nothing like a stressful night. So now I'm not in my cat room tonight. I'm in my husband's office. Whew. Okay. Take a drink of soda and I will calm down. My cheeks are red from the stress of,
Internet's stressful.
It is.
Very unlike you, Suzanne. I was about to send you a text.
I know. Actually, that reminded me, I need to move my phone because Matthew was texting me too.
Uh-oh.
I'm not sure if he made it on here. Hold on a second. We got a few other late comers coming.
He's here.
Oh, Matt is?
Yeah.
Okay. All right, good. Oh, and I see David made it here. So, all right. Somebody under the age of 18 has made it. So, all right, just give it one more second. I know we're starting to a couple minutes late, but again, with everybody straggling in with my chaos. So, I didn't mute my phone. That was my bad. And I hear their Instagram messages coming in, I'm sure. All right. Okay. We've got 33 people here. So that's a pretty good, because I normally try to cap it around 30, but I know that a lot of people are very interested in tonight's subject, and it should be a good meeting. So I'm going to go ahead and get started on this. Did that. Okay. I am recording this. This is not for public consumption. So this is not going to be broadcast out there, but there was a university person that needed to hear some of what we're going to talk about. So it's all for your benefit. So don't give away your PIN codes tonight or any big secrets. Please admire tonight's Snoopy t-shirt. I think we're on day 7 of 25 days of Snoopy, if you've not been following. Oh, and I have my Snoopy cup tonight also.
My dog is in a Christmas sweater.
Yay!
Oh.
Katie locked her dog in the other room.
She has a lot of energy. I didn't lock her, but she has a lot of energy, so she would only be distracting me right now.
But she is adorable.
She's cute. I can show a picture later.
Yes, we will do that for sure. Okay, nobody else is waiting. We're going to go ahead and get started. Thanks everybody for coming tonight. Sorry about the absolute chaos. You can thank Blue Ridge Cable. And because we live in Eastern Pennsylvania, we have no other internet service providers to choose from, so they don't actually have to make the internet work. So they've kind of got us trapped out here.
Perfect.
So tonight, I'm going to go ahead and I'm going to screen share to get us going. I have a tiny little intro thing as usual. For those of you that have been here before, you can just snooze through it. But for those new people, there's some information. And I do have a couple new pictures tonight to show you. So let me do this and let me know. Can you see my screen?
Mm-hmm.
All right. So again, welcome everybody. First meeting of December. I'm going to try to squeak in one more before Christmas because I do actually have two people that I've talked to about doing some meetings. One, Dr. Carrie Peters, wants to come back, and actually she and I are talking about doing a streaming thing from her greenhouse as long as we can socially distance down there. So that may be coming. And then I have another bug nerd friend of mine who has agreed to come on. So just keep an eye out for that. If I was a better person, I would have a regular, consistent timing of doing this, but life happens. And so I'm just happy to get these together when I can. So the rules for tonight, this is for growers only. I appreciate, and hopefully everybody here was honest with me about who they are. If not, you should feel guilt-ridden. And you will end up with hemp-russet mites because of karma. But anyhow, don't, record this or take images. And tonight we're just having a discussion, so there really aren't any images. And I really encourage discussion for those of you that have been here before. So I'm going to have a discussion with Katie, but if you have questions, let us know because we'll be glad to take them as we talk about things. For those of you that don't know me, my name is Suzanne Wainwright. I'm the owner of Bug Lady Consulting. I've been in business 20 years now. It's strictly a consulting business. I do get quite a few people from Instagram messaging me asking to buy ladybugs and things like that. I don't sell any insects. I'm strictly a consultant. I did go to school for this. I went to the University of Florida and I have degrees in entomology and environmental horticulture. And in normal times, I travel around the US, Canada, Caribbean, and help people with their pest issues. So first news update on Michael. For those of you that have been following Michael's journey, she's actually had a really good week. We had to have more blood work done last week because she has multiple issues going on. There's, some permanent neurological damage she has. That's why her tongue hangs out and she can't really walk in a straight line. But it is a little enduring. She also, they believe, has some kind of whoever just came in. I'm going to mute Theo for him. There you go, Theo. She also has some kind of, they think, immune disorder where she's struggling to produce enough red blood cells, but right now she's on a low dose of steroids and we're trying to wean her off, and hopefully that will help keep her blood cell count up, because last week she was in the normal range, but that is with being on medication. But she's eating well and she's a total Fanart stinker that's been just running around the house like crazy. So it's good to see her have good energy and she's getting along with all her other animals, which is super nice. So that's the Michael update, and I appreciate all you guys that have helped, because the vet bills have been north of $3,000 for her. And it's been super awesome how everybody's been able to help me save her. For those of you that don't know, she showed up in my backyard about a month and a half ago, incredibly sick and needed surgery. And there's been a lot of issues, but she's such a sweet cat. She's super sweet and super nice. So that's the Michael update. My other update, the other thing I've been doing is, if you don't follow my Instagram account for candy, I'd appreciate it because I could use a few more followers there. I do historic candy making from the late 1800s. And so this is during my days right now what I'm doing. Normally I volunteer at a bunch of historic societies here, but they're all closed because of COVID. But I'm still making my candies, and I'm trying some new things this year. So if you're marginally interested in candy history, you can follow me over on Instagram. I have a more active Facebook page for the Clear Toy Candy, where I've been posting some videos and things. But yeah, this is what I do when I'm not doing bug stuff on there. So that's that. So tonight is absolutely free, as always. And what I've been telling people is if you do get information out of it, I appreciate a tip for organizing this and dragging my friends onto this when they're after hours. I don't worry, when I actually get to see my friends again, I will buy them a beer for participating in the event, or bribe them with candy, one of the two. But if you do get something out of it, if you can leave a tip either for PayPal and Venmo, normally I would not be asking this, but I've essentially not worked now for about 10 months due to COVID and travel restrictions and everything on that. Hopefully by spring, I'm going to be back out on the road. I was hoping it was going to be this winter, but it's not looking so good right now. So tonight, yay! We have Katie Britt on, which I just love her to pieces. And this is one of the things I truly love about my job is, I work out in the field on a very applied-ish side of things. And I love working in conjunction with the researchers at the university so that we can combine all our brain power to solve problems. And, to solve a lot of our problems, initially we need just some down and dirty research, and then take that and, how can we apply that out in the field? And I'm so thrilled that even though Katie's in the vegetable lab, that she's been working on hemp and hemp pest, which a lot of that does cross over into cannabis. She and I've had a lot of in-depth discussions, and it's been really, really great. And I'm looking forward to see everything she's going to do in the future. But Katie, why don't you tell us a little bit more about yourself, where you're from, and just a little bio, please.
Yeah, for sure. So thanks for that really nice introduction. So originally I am from Marshville, North Carolina. It's a really small little country town. And if you are a fan of country music at all, it's also the home of Randy Travis. So that's a cool factoid about my town. I went to undergrad at Emory and Henry College. Most of you probably have not heard of that, but it's a small liberal arts institution in the mountains of Southwest Virginia, kind of near Bristol, if you're familiar with that area. But I majored in environmental studies. I didn't really know what I wanted to do with my life at all. And one summer I took a summer class focused on entomology and just absolutely fell in love with the subject. It just fascinated me, and I was sold on the first day. So that's what I decided to pursue a career in. So I graduated from Emory and Henry in 2013. And then in the summer of 2014, I started my master's in entomology at the University of Tennessee in Knoxville, Tennessee. The focus of that work was the kudzu bug, which is an agricultural pest of soybeans, so a little bit different than hemp for sure, but it gave me those initial skills with how to do research and introduced me, not introduced me, but let me get further into the field of entomology, where I, fell even more in love with insects. So then for my PhD, I took a few months off in between those degrees. I knew I wanted to do something with insects still, but I also wanted to get a little more into plant research. So it was during that time that I learned about hemp and found out how incredible of a crop it is, much like anybody else who learns about hemp for the first time. And I decided I wanted to work with it. So, I put some feelers out for professors that might be good to work with. And it's, life's a little serendipitous, I guess, but I ended up finding an awesome advisor at Virginia Tech who agreed to let me work on hemp and insect pest research, which at that time, I only knew of one other person doing that work. And, everything has kind of blossomed since with the legalization of hemp and everything. But because of that, I got my foot in the door early. We were able to get some of these initial studies out of the way so that, at this point, we're kind of a little more in depth with the research instead of just doing survey type work. So I started here at Virginia Tech in 2017. So this 2020 field season was my fourth summer of research, and I'm going to be wrapping up very soon. But it's been a really good journey. I've learned a lot along the way, and I'm sure Suzanne will ask some directed questions tonight, but yeah, we'll talk about it more in depth. And thanks everybody for being here. I'm really excited to talk to you all and share with you what I've learned so far.
Oh, cool. Well, my other cat has now joined us because he can't stand not to be involved.
Perfect.
Yeah, I know. Well, there's no door to this room, so I can't lock them out. So there's my contact information for people if you need my email address. Obviously, all you guys know where to find me on Instagram. So I'm going to stop sharing here, and so I'm going to go right in and go for the first question, because I know a lot of cannabis growers have been battling cannabis aphid, and which is forward-on cannabis, which I think there is a lot of confusion out there, because people think that when we're talking about cannabis aphid, it's just an aphid on cannabis, because there are numerous foliar species of aphid that do feed on cannabis. But in this particular situation, and what you've been working with and interested in hopefully doing more with, is this forward-on cannabis species. With that, and remember everybody that, she's been working mostly on a field hemp kind of work. So if you're an indoor grower, you have to think about the transitioning of the differences between indoor growing and outdoor growing and how it applies to your situation. So think about that, and that too, when you ask questions too tonight, just say, oh, I'm, a cannabis grower and I've got cannabis aphid indoors. How do I get rid of it? Well, Katie's been working again more hemp outdoors-ish, but I think there's been some stuff in controlled environment. So just remember where everybody kind of works. But that said, so why the cannabis aphid?
That's my first question. Yeah. So why? Yeah, it's a great question because, speaking from an entomology and a kind of insect research perspective, we know, and I'm not over exaggerating here, we know next to nothing about this insect in this particular species. So, everybody here, you're probably familiar with the history of hemp, period. It's not been legal in the United States, but, maybe within the past 10 years, depending on what state you're in. So, almost every other insect, no, that's not true. But a lot of the other insects out there, we've had years and years and years to look at their life cycle, their behavior, their relationship with different types of crops. So we're kind of really behind here with cannabis aphids. So not only have we not been able to study the insect, but we've not been able to study the particular crop in which it conducts its life cycle with, So where we are now, we know that it's present in hemp, cannabis, whatever you want to call it. Some awesome work has been done by Dr. Whitney Cranshaw and his former master student, Melissa Schreiner. So they're from Colorado State University. And I would wager to say that they probably know the most about this insect. As Whitney's student has done some life cycle work, general biology studies with this insect, trying to rear it on different types of crops, other than hemp or cannabis, trying to see if it can conduct that life cycle on other plants. And what I've gathered from them and what I've seen on my own is that basically it seems like it's only living and reproducing on cannabis. So that's a big deal, right? Because if this insect is present in, anywhere in the vicinity of your hemp crop, it's going to go exclusively to that crop. Okay, let me think some more. Why cannabis aphid? Yeah, that's the main thing. It's a specialist. It only feeds from this type of crop. So again, if it's present, it's going to feed from your hemp. So we really need there's a lot of more work that needs to be done, but we've got to get to the point where we understand the relationship here and what it's really doing to this crop, and given that it's a specialist, maybe it doesn't wreak as much havoc as other pests might within the hemp system, but we're still in a state of learning about that insect right now. Do you agree?
Yeah. Well, first of all, just, I did not let Katie know any of the questions I was going to ask her.
We're on the fly here. I'm getting nervous.
Don't get nervous. Everybody we're all good here. My gosh. I'm running from room to room in my house with cats on me. We're all good here.
We don't bite.
Yes. Yes.
No, I appreciate all of you. I'm just excited to be here.
I would agree with you. I will say I do believe that cannabis aphid can wreak a lot of havoc on indoor grows. I think one of the scarier things for me is what we've kind of seen about how it can overwinter on cannabis plants outdoors or hemp outdoors. Because Whitney and I have had a lot of discussions about this, because with I think this is concerning to cannabis growers is if you have hemp growers and anybody's growing hemp and goes to seed and you have hemp that escapes to the wild, and now you've got volunteer hemp plants growing around and cannabis aphids get on those, they, can overwinter, lay their eggs, overwinter on the plant material. And then next spring, boom, the aphids are right back and hop right back into the crop or back into your field. Where with most aphids, we see an alternate host for overwintering and not its actual host plant for overwintering, which is something really interesting to me, that it has the ability to do that. And again, if you have a cannabis grow and you have people growing hemp outside or near you, because if you're growing hemp for fiber or things like that, you probably have a higher tolerance for pests than you would an indoor crop. It's just like, with ornamentals and vegetables, every leaf has to look perfect. And with vegetables, as long as the vegetable looks good, who cares what the leaf looks like? So we definitely have different pest thresholds. But I'm getting contacted pretty much every day. Either somebody's got forward on cannabis, or they've got root aphids every single day. And I'm convinced it's an all lower 48 at this point. I'm sure it's going to end up in Hawaii too at some point, with someone taking cuts over there and up to Alaska. But I don't know. Well, we know it can overwinter in Colorado, but I don't know if they know yet the northern limits of it overwintering.
It overwintered in Washington.
But Washington's not that cold.
Yeah, true.
I want to, will it overwinter in Alberta? If it can survive Alberta, because I've been to Alberta in winter, I almost didn't survive. So, but Kate, so have you, with your work for this cannabis aphid, what have you been looking at stuff? I know you were going to do a survey, but with COVID, things haven't gone as planned with that.
Yeah, so that's a good question. One of the things I have tried to do is I'm working with another graduate student who's in Canada. He's in Ontario, so just north of the United States border. But we're trying to, we're reaching out to entomologists in every state within the continental U.S. and Alaska and Hawaii. So every state in the United States, basically. But trying to see if we can get samples of this insect so that we can confirm its presence in every state. And like you said, Suzanne, I think it's everywhere. We haven't documented it, but I think, since it needs this crop to be able to survive and reproduce, this crop is pretty much everywhere now, or it has been grown historically. So there's going to be a resource for it to live and breed on. Now with especially hemp being federally legal and some form of cannabis being grown in every state, that gives it the potential to exist in every little corner of the United States and Canada now. So I think it's going to be everywhere and going to continue to spread. And, especially if people are purchasing plant material from another state, bringing it in, that's just facilitating the spread.
Yeah, because you can watch as legalization happens, how it's been moving. But again, it's I'm sure it's in the black market too. But
Sure.
so would you want samples of Forward-on aphids from growers sent straight to you, Katie?
Yes, I would appreciate that. And I can give you a mailing address in which to send those. But, one other thing that's making me think, even if you see aphids on your crops that aren't cannabis aphid, that's just as much worth identifying as cannabis aphid because, I think that's another interesting concept. I don't know that much about this insect, but it seems like rice root aphid is pretty prevalent everywhere. I know that's one I know almost nothing about, but it's presenting a huge problem. But I mean, that combined with other generalist species like green peach aphid or potato aphid or other things like that are starting to be documented on this plant. And we need to know just as much about those with hemp as we do cannabis aphid.
Yes, that's why I've had people sending me samples, but I think sending you samples would be excellent.
Yeah.
So Katie will provide an address. Don't send plant material. Just put the aphids.
Yeah, we don't want anybody to get in trouble.
Yeah, and just put the aphids in alcohol in a sealed container because I've had people send me samples and it gets here and there's just a dried up container because the alcohol has evaporated or leaked out. Those little 1-2 dram vials are excellent for sending aphids, and some of them have Nalgene seals in the top. I buy them by the bajillion from BioQuip, and that way I have them and carry them all the time out there. So, okay, she put the address to ship them to. All right, there was a question in the chat. What's the life cycle of the aphid when it comes to reproduction and best approach to defense and extermination? Well, they must exterminate. I mean, when it comes to the biology, the biology is, I would say, pretty straightforward. I think where it gets a little wonky is, normally in greenhouse and protected culture, we never see egg laying. We don't see males. And with this Forward-on aphid, under right environmental cues, it will start laying eggs indoors, which is what is so unusual about this. I actually say it's a good thing because since the eggs don't get a chilling period, they won't hatch. And so it's kind of like, the female aphid's expending all this energy to lay this egg. That's a dead end instead of, having live babies. Even though what's weird is I see females right next to each other, some having eggs and some having babies. So it's weird. It's not like it's 100% cue that they all start egg laying. And I don't know what that's about. I don't know if there's different biotypes, that's a possibility. And when I say a biotype in the insect world, is kind of like a cultivar where it's the same species, but there's enough difference between them that they kind of break them out. And we see that can be from behavior, from pesticide resistance issues and things like that. But it's possible, there's been more than one biotype of this ape, because it's kind of weird because usually when you get an environmental cue, they're all like lemmings and everybody goes and does it, in the insect world, and to have only some do it and not the other, it's kind of weird on that. Do you have anything to add to that, Katie?
No, I think that sounds good. I was going to say, I don't know the life cycle of this insect that well, but that's what I was kind of trying to point out earlier, is that we're still learning so much about it in general. I can speak from what I've seen outdoors, though. From my experience, you can see them in rather high numbers, throughout the season. But the time of year that I most frequently see them is around the fall, right before the plants are going to be harvested, which, like Suzanne was saying earlier, that's, a really important time to be checking whether they're there or not, because if you don't get rid of that crop material or that crop debris after you might harvest from your outdoor grow, if that material is left there, then that's going to give them the potential to have something that they can remain on throughout the winter. They'll survive throughout the winter, and then they're going to be there in the spring, ready for whenever you plant during the growing season. So that's kind of important.
Yeah, because that's where we've seen it, and growers reported back to me is they find the aphid in the compost piles.
Yeah.
And I've been to enough cannabis facilities where they have these huge compost piles outside, which it's just a harbor for insects and disease. You gotta make that stuff go away. And by the way, everybody keeps typing questions. You can just, turn on your thing and flag us down to ask for questions, because it's hard for me to, it goes dead air silence when I have to go read on that. And right now tonight, we're going to skip really talking about rice root aphid, since Katie hasn't been working on that. Yeah. And I don't want to say we've beaten the rice root aphid to death in other meetings, but we've had a pretty significant discussions on those. But we can always sometime come back to those. Do you ever see some parasitoids on greenhouses? Okay, so there are definitely parasitoids of this aphid and multiple species of it. And that's what's a little, I don't want to say completely confusing, but a little confusing. And we're seeing different parasitoids. So far, it seems that different regions of the country have...
I mean, do you want to see some?
Oh, sure.
Suzanne.
Oh, I can read. Yeah. So,
looks like we have one parasitoid right here. Let's see if it focuses.
That's right, because we were just talking about this, weren't we, about 2 weeks ago?
Yes, we were. We're pulling the greenhouse right now, actually.
Okay, save those. Yeah, save those parasitoids. They look like little brown balls, for those of you that haven't seen them before, because we're interested.
Can you see that at all?
I have to kind of lean in, and you guys can look at my hairline.
Go a little further.
I can see by your thumb.
Go a little further away.
I have terrible connection down here.
At least your connection's holding. I got dumped like 6 times earlier tonight.
Right, I might be able to find a better example. This cultivar is like an absolute aphid haven.
Yeah, because that's something we're interested in, is knowing which parasitoids are working where, because commercially there's a lot of parasitoids available, and I want to make sure my growers are not buying parasitoids that are the wrong ones for their growing situation. But we haven't been able to exactly nail down, because we've seen, whoever 817668 is, what's interesting in California, here, we'll look real quick. I don't know if you guys can see his.
There's 2 just on this one leaf here. You can see.
A little shiny, yeah.
In California, where they've collected samples, they've found Ophidius colmani, but Whitney and his student has found a lot of Irvi, where up in the Niagara area, one of my growers had really good success using Matricaria in the fall. So this is what's kind of weird. Also, and this is a whole nother discussion, if you're buying parasitoids, there's differences between the different companies. Someone I know has been buying product from the different companies and QCing it to look at emergence. And then also, not only can the parasitoid get out of the mummy that you buy, but can the adult then fly? Because they have to be able to get out of those gold balls and fly. And when you buy product, there is some stress involved with shipping and moving and everything like that. So if you are buying, let's say, colmani and not seeing success, then I would just check the quality of it to see what kind of emergence you're getting. And then even hold some in a cage to make sure that they're flying around in the cage before you let them go, to make sure that they're doing that. So on that, I'm trying to leave my phone. Oh, can I let me in? Can I let who in? Oh, hold on a second. Admit. Okay. I've had great success with green lacewing. Green lacewings definitely will feed on them. I know, Katie, have you out in the field seen green lacewing larvae when you're out in the fields looking around?
Every now and then. Normally what I notice with lacewings more is the eggs. I see the eggs that they lay on plants, and I'll see adults every now and then. But the main thing So this is one of my favorite things to talk about with cannabis aphid. I'm just going to go into it, but outdoors, it's amazing the activity that you see from lady beetles coming to consume cannabis aphid and help you manage those populations. I haven't written this up for publication or anything yet, but I did do an insecticide trial with cannabis aphid outdoors, and I think it really speaks to the power of lady beetles as a natural enemy against cannabis aphid, because, you know, I didn't really see, I personally, and, you know, looking at the data, couldn't see marked differences between the treatments that we used, but what we did see was a drastic reduction in cannabis aphid populations just due to the lady beetles. So I think outdoors, you know, you do have that benefit, whether it's lady beetles or maybe green lacewing larvae if you see them more often, but natural enemies really help manage that insect outdoors.
So before everybody goes and runs out and buys buckets of ladybugs, who I will have to disown you if you do that, by any chance, are you guys keeping track of which aphid species you're finding by any chance? Because I would think Harmonia would be the one that'd be kicking ass in there.
Oh, us. It's mainly that one, and you know, it's just whatever we see commonly outdoors. What I'm trying to say, the invasive one.
Well, the Harmonia is the Asian multicolored ladybird, but that's super aggressive. That's the one that gets into the soybeans and decimates the soybean aphid and everything. And then one of our native is there's the Hippodemia conversions, and that's a little more oval, and it's got the commas, and that's the one that's wild harvested out of the Sierra Nevadas. That's what's sold, but again, they're wild harvested out of the Sierra Nevadas and shipped all over. But if you've been around me, you know that we know they carry parasitoids, they carry a disease called microsporidia. You know, there's lots of issues with those wild harvested ones. But I'm curious to see too, do you know there's the pink spot ladybird, Comia maculata? That's another one I would think, because I haven't seen yet a good outdoor hemp aphid infestation on hemp to be able to go out there and see. Most of the hemp I've seen have had light other issues, but not one of these bad infestations, because it would be really interesting to survey which particular ladybirds, because their diets are very varied. Like the Comia maculata, the pink spot, like she requires a lot of pollen in her diet. But she, you know, it's interesting. I did a bunch of work with her, them males and females, back in the late 90s, and believe it or not, the larvae were really good on spider mites, but you don't see much documentation about that. So, you know, people, I don't think all the ladybugs have been studied as much as they could be, and also to see if the adults are there, are they egg laying? Because I've seen it indoors, but that's because they've kind of been forced to. But, you know, outside in hemp, are ladybirds coming in and saying, yes, I think this is a good spot to put my eggs and lay her eggs, you know, there on the crop. So,
Suzanne, is there any way to get, I'm sorry.
You just scared me.
Is there any way to introduce ladybugs other than the convergence? Because those are the only ones that I can find commercially available.
Well, BioBest does sell, I think it's the twice-stabbed ladybird. The problem is you're going to look at that and you're going to like have a stroke over the price. Sure. That's what happens when you go from being able to go out and vacuum your crop up to actually growing it yourself. I mean, it would be like if, you know, you could pick weed off the roadside and sell it compared to you cultivating it, your cost goes up. This is why so many people have gone to lacewing larvae, because they're relatively inexpensive. They kind of do the same thing, but you don't have the benefit of the adult. But I will warn you with ladybirds, and I've got enough pictures to back this up, they will get stuck in bud, and nobody wants to pick ladybugs out of bud. Now, there is one other, and I've been pushing somebody to do this. So this Comia magilla, the pink spot ladybird, which is another one of our native. So they've actually developed an artificial diet for it. And so you can rear it on basically baby food. I'm just going to call it that, but it's a mush designed for ladybirds. And the company Insect Lore bought that technology, and they actually sell pink spot larvae for kids' science kits. And I've had numerous emails years ago, like, can you guys just go into production and sell them to growers? And they said, nope, that's not our market. They want to sell butterfly rearing kits and bug goggles and insect posters and kid stuff. But and thing is the diet, a lot of that was developed with funding through the USDA. So that information is out there. It's not top secret information. And so I'm still waiting for somebody to decide, wow, I want to start a ladybird beetle rearing company.
Yeah.
You know, because I do think that with the artificial diet and the technology, the price could come down and now especially all this happening. And before, when they were rearing them down in Florida, they actually had two different ladybirds of the CMAX, one for West Coast of US and one East Coast, because they knew they were different. So they were a bit targeted too when they did that. So, I mean, that's the best kind of long answer I have for you right now. Again, BioBest has the one ladybird, and there are some other specialists out there, but they're not aphid feeders like Delphastus and Stethurus and stuff. Those are very specialized ladybirds that are not what you want. But all the other vegetable growers and ornamental growers somehow magically grow without ladybugs.
Yeah. Yeah. And so it's not commercially used by anybody but cannabis, basically.
And home gardeners that think they're doing the right thing. But yeah, we thought that the ladybug harvesting industry was just going to kind of choke itself out and die. But nope, here come the cannabis growers, and they're going to resurrect one of the worst environmental things possible.
Yeah, that's our MO. So I did before I knew all that and started working with you, I also did a release of the convergens, and I released them in my large full season greenhouse, and that was cotton melon aphid, and those died back. And like 6.5 months later, we're having a different cycle in there, and the hemp aphid popped up. And then shortly thereafter, there's all of a sudden this explosion in ladybug population without having introduced them for months and months and months. And the population sort of grew in lockstep, although the ladybugs never did what I would consider effective control. But I was just amazed. Me and my staff were amazed by how many like larvae we were finding and egg clusters and everything. I didn't think to confirm that it was the convergens, but I just assumed that it was. I don't even know if we have any native.
Oh, of course you do.
Okay.
Of course you do. And this is where, you know, researchers have to get more into genetics, because even if it was convergent ladybird beetle, is it the same population? Because I mean, we do see in greenhouses and protected ag that have all this biosecurity, beneficials do get in there. And if there's food, they will survive. But if it was the same population, that tells you you've had enough food in there for them to sustain them all this time, which I'm not sure with the Hippotemia convergence. It's pollen requirement, because like I said, I know the CMAC requires it in their diet, like Aureus has to have it and stuff. So, and you have to wonder about an indoor grow because there isn't pollen in there. So my guess is maybe they don't need pollen in their diet, possibly. So that's a whole nother rabbit hole to go down. But yeah, when you have pest population rise, the predator is going to come, but usually the predator never eradicates the pest, because it wouldn't make sense to completely eradicate all your food source for all your kids. So, but it's probably not providing enough. You're still going to be above the economic threshold of, you can't, it's causing too much damage to the crop.
And I should abolish thinking of buying convergens as a control.
If you ever want to speak to me again, yes.
Copy that.
No, I mean, I don't want to say I would never recommend it. There may be a situation, but I would much rather, look at some of the other options of, initial knockdown spray, and then using combination of green lacewings and parasitoids. For now, we're probably going to know more because of people like Katie down the road who will do more research, and that can, put the cannabis aphid in all the cages and then release the parasitoids and watch and see what happens and then document emergence and all this kind of stuff. So we'll know better. Because I actually had a call with one of the biocontrol companies last week, and they're struggling because they just, you can't do research in commercial growing facilities.
Right.
There's too many moving parts. Sure. And you just really can't.
That's what I was going to ask when you said about checking on emergence levels from like commercially.
Well, that you can do. And I mean, the quick 30-second thing is you buy zip up cages for like $6-7 from BioQuip out of California. They're these little cube mesh cages. You buy your parasitoids, you dump the mummies in a petri dish, and then you zip it closed, and then you wait a few days, and then you'll see the parasitoids flying in there. And for this, you don't have to kill them. Like some of the other quality control stuff, you have to kill the stuff to, do counts in this. But you can either just kind of eyeball it and be like, yeah, that looks like there's 500 in there. And then you can take your cage out to your grow room, unzip the thing, and let them fly out. Nice. Or if you really want to count them, there's a thing called an aspirator, which is basically a handheld bug vacuum. And you can suck them up and you can count them 1, 2, 3, and then you know exactly how many. Or you can take like a quarter of the mummies and put them in there and then just do the math on how many actually emerged out of the percentage.
Because that's one mummy to one wasp, right? Mm-hmm. Okay. Yeah. I did a release of Clomani Irvi mix from Beneficial, and I felt like, because the Irvi are like outnumbered like 10-1 or something like that in that mix, I felt like the Irvi was what was effective because of the level of mummy population that I was seeing after a couple weeks of checking on it was really, really low. Even, we haven't introduced them in, 2 months now or something like that. And I'm still seeing the mummies, but in like tiny, it'll be like 1 in every, 20 plants will have a mummy on it.
Right. And that's what's good about the parasitoids, because they'll go find one where some of the predators need really high densities in order to work on there.
Right. Gotcha. So. Awesome. Thank you.
Thanks. So, Katie, do you have anything else do you want to talk about? You started to talk about some spray trials, didn't you, for cannabis aphid?
Yeah, but the main thing I wanted to point out, because I did that trial outdoors, is that, there's enough natural enemy presence out there that I think that, those insects, those predators will help you manage that population. Like, don't jump the gun and spray if you see, because I got one high level infestation in the field. I haven't seen that level of infestation since this one happened last summer, but still, from what I was able to see, the predator insects did more work than, I don't want to say the insecticides didn't work, but the natural enemy predator insects came in and managed the population. So don't just jump the gun right away and spray, because you may not need to do that.
Did you see syrphids out there feeding on them by any chance?
I do see a lot of syrphids in hemp. As far as, larvae feeding on those, personally haven't seen that as frequently, but that doesn't mean that it's not happening.
Because if you guys don't know what syrphid flies or flower flies are, make a little note, Google it, look up their larvae, because I've had several growers this year contact me, think they have caterpillars, and they're actually syrphid fly larvae, which are predatory on aphids. I've had ornamental growers. Oh my God, one of the worst things in my life was I had a hydrangea grower, and they had caterpillars on the hydrangea, so they sprayed it with 7. And they were these syrphid fly larvae, which were controlling the aphids for them on the hydrangeas. So it's, again, Paul, I'll go check and see if that's right. So it's, you got to make sure an ID, and, right now, I can help you guys with ID because I'm not traveling. But once, I actually get back to work, I'm going to be a little harder to get a hold of because I usually travel every single week. But for right now, I'll be glad to help you with ID. And Katie will too, right?
Yeah, totally.
But yeah. The aspirator in the chat, that's the one that I use and leave with growers quite a bit because it's super easy to use, inexpensive, and you can collect insects without damaging them. Oh, thanks, Katie, for putting the link in.
Yeah, it's got some pictures of syrphid adults and larvae on plants.
Yeah. But, and this is why it's so important to ID before you go out and kill, things. I can't say that enough. But, again, you guys on the inside, it's tough because, you put up walls and that excludes beneficials from coming in. And, even some of the greenhouses that have roll-up sides, we see pretty early in spring, the syrphids moving in. And also this 2 years ago, I was really surprised in early spring in Pennsylvania, I was actually finding aphidolytes in a greenhouse lettuce crop coming in from outdoors. Aphidolytes is another one that's a predatory midge that feeds on aphids, but I'm not sold on it for cannabis. So don't get too excited about it because I think it takes too long for it to work. You've got to build these releasing love shacks. You kind of have to pre-feed them, pre-mate them before you release them. And you can't use in conjunction with some of the predatory mites because the predatory mites will eat their eggs before they even hatch. So it's kind of a complex thing for that. Katie, is there anything else about cannabis aphid you want to talk about before we move on?
I can't think of anything factual that I have to contribute, but I did want to say, I know it's really hard to be a grower with this insect right now because we know so little about it, but just emphasizing again that, me and other people within the university system are working on this and trying to learn as much as we can within the United States. So eventually we'll have more to share. I can also share a link to some publications that I've contributed to along with Whitney Cranshaw. So I'll put those in there and make sure you all have access to those. It could give you more information in a pinch.
Hey, you guys.
Oh, hey, goat people.
Hi, Suzanne. Hi, Katie. I'm just going to go through my questions here. Looks like we already answered that. But you don't have the answer. What temperature will kill the overwintering aphids? We don't really know that. We hit about -20 here for a good 2 weeks. I think we're zone 5 up here. So, yeah, we're zone 5, -20 Celsius. And I don't know because prior to knowing that we even had the cannabis aphid, we did compost some leaves, and now I'm super worried about it. The other question, can the Fordon cannabis survive on anything other than cannabis, or does it depend on the cannabis for its life cycle, or will it thrive on other things?
So what I've learned so far, like I was saying, mainly from Whitney and his student Melissa, and then a friend of mine and Suzanne's, her name's Emily, up in Vancouver, British Columbia. They've both done some work with this insect, trying to rear it exclusively, on cannabis and then other types of host plants. And what they've seen so far is that it's only cannabis that's allowing Fordon cannabis to survive and reproduce. So, based on the studies that have been done right now, it does need cannabis to complete that life cycle. Although we kind of speculate, there has to be some other type of host plant that's keeping it going. But again, based on what we've seen right now, what's been documented with research, it's only cannabis species.
So how does that happen? Like, is it a mutation in say a green peach aphid or something that then makes it a cannabis specific strain of
it's like goats and horses. They're complete. Well, I mean, okay, let's say one is a thoroughbred and one's a Clydesdale. They're completely different species, but they are the same insect.
Yeah, that's a tough one because we have all these species of aphids that they might look alike, except for certain colorations or what they feed on. But yeah, they are just as different. I feel like this is a stretch saying this, but they're basically just as different as we are from a dog because we're completely different species. Do you agree with that, Suzanne?
Well, I mean, the different aphids are, I would put them in as like different breeds of dogs because there's enough similarity in anatomy, and I mean diet's kind of iffy too, because again, you can't just take a cannabis aphid and go stick it and it will eat something else. I mean, they are not omnivores. They are very, very, very targeted, and even physically trying to Emily, our friend we were talking about, that's up in BC, which by the way, she did get a job. They did move. But anyhow, she tried to move cannabis aphid onto hops and hop aphid onto cannabis, and it was just a big no-go. And physically, you can't tell them apart, but when you get them under a microscope, their hairs on their body are actually shaped differently. And so they may look similar to the naked eye, but they really aren't. And it'll be interesting to see too, because hops aphid has alternate overwintering hosts, because it doesn't overwinter on hops. And where the cannabis aphid is staying on the plant. And yeah, I know Whitney's been looking for another alternative host, but it would have to be something so close. But I just think there's been so much weed grown everywhere. And for the most part, when it was black market days, people were just keeping the numbers so suppressed with orthene, durazban, imidacloprid, and all that stuff. But there were small pockets of it around. And now, since the legalization, those products are not being used, and you have way more plant movement, it's just allowed this cannabis aphid to explode, because I think it's been here for a long time.
I completely agree. Yeah.
Just
low, illicit grows or the ditchweed that we mentioned earlier is what's kept this population sustained until now, which we have wide scale production.
Yeah. Because I guarantee you there's lots of abandoned patches of cannabis around that, this aphid's been in and has been able to, survive, because people do plant it, and people sometimes don't get back to it. So, yeah. Do you have any other questions?
There's a question about heat treatment. Oh, go ahead, Sarah. Sorry.
It's okay. Suzanne said, "Oh, thanks for putting that up there, Katie," but I didn't see anything come up. So I'm not getting the links that you're putting up.
I'll repost it. Yeah.
Where am I? Well, should it come up on the screen?
There's a chat bubble at the bottom of your window.
Oh, it's in the chat. It's in the chat thing.
Yeah, I only put one in about syrphid flies so far, but I'm going to paste into publications that mention cannabis aphid.
Okay. Yeah, this summer I was one of those people that sent the pictures to Suzanne with the syrphid fly on our outdoor, and it was not the cannabis aphid, but I had never seen anything like that. And then I started hearing stories from all over, high levels of them. And yeah, tons of syrphid flies around this year, or the larvae and aphids in outdoor situations. And I've just never seen them before, and I'm quite observant, but this year they've exploded, which tells me that the aphids are exploding to support the syrphid fly.
And I would recommend, I'm not one for, companion planting and all that as much, but I definitely, from observations on my property here now for almost 20 years, if you want those syrphid flies outdoors or near your grow, you just plant things like yarrow, Shasta daisies, and tend to try to go with older varieties and cultivars of things, because a lot of the newer stuff has been bred for human aesthetics and not have pollen in them. And that's true with a lot of the echinacea plants. Like these new echinacea plants really aren't these hot pinks and all these cool colors. There's not pollen in them for the beneficial. So they're pretty for us, but they're kind of useless as attractive plants. So I try to find the oldest, older varieties that haven't been tinkered with so much that the insects really like. Calendula is another one that I really love to grow a lot of. And so that is another one. And again, I figured these plants out just by going out and observing and seeing what's going to what plants. But definitely yarrow's an easy one. And again, they like the big open flat flowers like Shasta daisies are really good for a lot of those flower flies. So. All right. I saw someone ask about heat treatment. There's not really been any research done on cannabis aphid and heat treatments yet. I was having a discussion with a grower over the weekend about heat treatments for fungus gnat eggs. And actually there is a little work on that because since fungus gnats are an issue in other crops, that's why there's research that's been done on that. So there is some heat treatment stuff. The hardest part is even if you have a room and let's say you can heat the temperature in the middle of the room to like 120, is the corners of your floor going to be 120 under, underneath the bench, under the leg? Are you going to get to those temperatures thoroughly in there to heat treat everything? And that's been a challenge for that. And in this particular situation, eggs are much harder to kill. So if you just have adult aphids, it would be easier to treat them with heat than it would be for eggs because eggs are designed as to survive tough conditions out there. So we just don't have a lot of information on heat treatment yet, for sure.
What about cold?
Well, but if it's instantaneous cold, it would kill them, but it's probably going to damage if there's no plant in there. But if it's a gradual cool down, that's when they'll start laying eggs because they're preparing to overwinter. Okay. So, but again, how cold can they take it? I mean, Chaz, I hate to bring you up, but I think it was Chaz that saw them like in midwinter walking around in a compost pile. So cold's not, that was you, Chaz, right?
Yeah, that was me. It was like a 2-week cold snap in Washington where it was probably, it was well below freezing for a long period of time. And then there was still cannabis aphid crawling around in the compost pile even after that long cold snap.
Gotcha. So we harvested out of that house that I was describing, and like a week straight, and our overnight low was like 18, but it sounds like that's probably not going to do nearly as much as I thought it would.
Was that Fahrenheit or Celsius?
Fahrenheit.
Okay. Sorry. Just always have to, make sure on that. But and this is what will be interesting because now since there's more hemp going over to this winter to see where it is next spring to know if it overwintered on the outside. Because, previous years people haven't been real observant of this. But now we'll know more from that. So.
Right.
Okay. Are there any other, because not that I wouldn't chat about bugs all night, but I had to have my husband give me a blanket. I'm freezing. I don't know why I'm so cold, but I'm freezing.
I had a question about, Katie, I'd assume that these will be vectors for diseases also, like the hemp latent viroid, which although I see the pest as an issue, I see that being a much longer lasting issue that can drastically affect people's production.
So you're asking about cannabis aphid and pathogen transmission?
Yeah.
Yeah, that's a really good point. And, that's something we haven't seen anything like that yet with Fordon cannabis. Not to say that won't change in the future, though. As this becomes a more widespread produced crop on a year-to-year basis, it's here to stay pretty much. So seeing how this insect's relationship with the crop might change in the years to come as the acreage continues to increase, it's something we're going to have to keep monitoring for sure. But at this time, I don't know of any reports of Fordon cannabis transmitting any kind of plant pathogen. Suzanne, yeah.
I haven't seen any either. And aphids can vector virus, but they're usually not the big guilty party. If you, had to say, word association, virus, I would say thrips, because generally thrips are more of the virus vector. And, even when you look at, the beet top curly virus, that's one species of insect that can vector it. It's not even, I can't remember if I think it's a leaf hopper, not a plant hopper. I think it's a leaf hopper. Yeah. It's one species. It's not even all leaf hoppers. And you have to make sure it's that one leaf hopper. And it's only found like west of the Mississippi. Yeah. So I've not seen any correlation with virus and insects at this point. A lot of these viruses can be transmitted mechanically. And that's more likely, I think, maybe what we're seeing. Hopefully, because Purdue's been doing a lot of disease work. Again, I can't say enough good things about that DIG conference that was, I don't know, about 4 weeks ago. Katie and I did. Yeah. The disease talk on hemp cannabis there was unbelievably good. I mean, she went through, she's had stuff I'd never even heard of before. And she had pictures and a lot of, I'd worry about this. Don't worry about this. Yeah, we're seeing this. It was really good. And I've kind of pushed for her, Jenna, to speak at the Cultivate show this year on the diseases again, because I already heard the talk once. I want to watch it again because those that attended the conference, you can rewatch stuff because it was really good. And I'm hoping Purdue will do more virus work because there is a big gaping hole in that. So
who was that presenter that gave that?
Jenna, what's Jenna's last name?
Beckerman. I can type it in the chat too.
Yeah. She's one of those internet stalker. I mean, not internet stalk her. She's not an internet stalker. So stalk her, follow her.
It's a directive. Yeah.
Yeah. Yeah. She's I don't miss hearing her speak. There's certain people that I never miss speaking, and she would be one on my list now.
Awesome.
So having another shout out to another plant pathologist, since I'm in kind of the southeastern east range, Nicole Goche from University of Kentucky's also done a lot of stuff too. And similar to Jana, I think Nicole's got a lot of extension pubs that you could find on the web as well. But I'm putting her name in the chat.
Thank you for typing because I'm doing a bad thing. I'm holding my phone watching eBay right now.
Multitasking.
Yeah, of course, So I know there's a lot of things about aphids, but I kind of do want to move on because we've kind of spent an hour and, of course, we'll have more talks down the road to talk more about stuff because I kind of want to talk about defoliation. And maybe when I put my little blurb up, I should have said defoliation as it applies to pest management, because by defoliating plants, she, as in Katie, they're what's the word I'm looking for? They're basically trying to make the plant think an insect ate their leaves, and how that affects plants. So why don't you talk about your research and the work you've been doing on that, Katie?
Yeah. I was going to say I could pull up a presentation, but I can just kind of talk through it. So I've done some studies with simulating defoliation on hemp. And so this is trying to simulate chewing type of insect damage on leaves of hemp. So if you've grown outdoors, you're going to be able to notice that there are a lot of leaf chewing insects present in the crop, such as beetles. Some of the caterpillars will feed from leaves, mainly beetles and grasshoppers, though. So one of my big goals was kind of to figure out if these insects and their type of herbivory on the crop would lead to any kind of negative yield impact. So what I did was, set up these experiments in the field on a replicated plot design and used shears or kind of plant clippers to clip away certain percentages of leaf area from the plant. So with these studies, I simulated this damage at certain times throughout the season to simulate the damage at early, mid, and late season within the growing season. I'm going to sum it up for you really quickly and say that when analyzing the data after taking yield at the end of the season, I was not able to see any kind of differences in yield from this type of injury. So I know that, simulating this injury with shears is not exactly the same as if insects would have fed from the crop, but from surveying the literature and looking at defoliation studies that have occurred in other types of crop systems, this lines up exactly with what other researchers have done in varying crop systems. So because of that, I feel really comfortable with the way I did this study. And again, when they simulated this damage in other types of agricultural crops, they were able to see negative impacts. And so me doing this, the exact same way in hemp and not seeing any kind of negative impact really makes me feel secure and comfortable with this methodology that, I wasn't seeing negative impacts to yield. So I think that speaks to the fact that hemp is a pretty resilient crop, that it can withstand a lot of, damage to leaves or loss of leaf area, and the yield's not going to be negatively impacted. And I want to say too, I did these studies in grain and CBD variety hemp, and it was the same on both ends. So physical yield was not affected. In grain, that physical yield was seed production or seed weight per plant within these plots. And then in CBD, that was bud weight with the plants that I defoliated. And in addition to the physical yield, so bud weight or seed weight, I also worked with a commercial lab to do HPLC analysis to see if cannabinoid content was altered due to this damage as well. And I saw the same type of results with the cannabinoid values as I did with the physical yield, that, there may have been slight variations, but overall no real differences in cannabinoid content due to these levels of crop injury that we simulated. So again, things may be different if, I put actual insects on these plants in the field to simulate this damage. But like I was saying, due to what I've read in the literature, I feel really comfortable with this, and I've not been able to see those differences so far. So let me supplement what I'm saying about my studies with what a colleague has done. So a colleague from the University of Maryland, Eastern Shore, simulated, I don't want to say simulated, but he conducted this study where he actually put corn earworms on buds of hemp in the field. And so the way he conducted the study, though, was that he put these worms on the plants, and then he clipped the buds that they fed on and tested the buds immediately after feeding. He was able to see differences in cannabinoid content, but that was only when the infestation level was really, really high. So I want to say that, the levels of worms that he put on the crop is higher than what I have seen naturally occurring in the field. And, he tested these buds right after they were fed upon. So, I think if that's happening, if you are, somehow getting your buds or getting cannabinoid values tested, right as a worm is feeding from it, there may be the risk that the CBD or THC content would be elevated. But, I think what I'm being able to draw from my study compared to that study that the guy in Maryland did with earworms is that I think maybe there is the potential that cannabinoid content will stabilize throughout the season, as that feeding occurs. If there's a gap in the feeding and when you're testing these cannabinoid contents, prior to harvest, I think it's going to stabilize out elevated when you test it after harvest, as if you tested it right as the insect was feeding. So I know that was kind of a lot, but if you have questions, definitely let me know.
Go for it, guys. Go.
I had a question about, so have you done any studies or trials with using salicylic acid to trigger the systemic acquired resistance that you would sort of get from, I assume that you would get from defoliation?
No, I've not done that. This has only been, simulating this type of chewing insect damage on plants in the field.
Gotcha. And what percentage of defoliation do you use when you try to do that?
Yeah, that's an excellent question. So the values that I chose were, 0, so that I could have a control amount, 25% of leaf area removal, 50%, and 75%. And for the record, when I, in addition to just simulating this damage, I also collected leaves that were defoliated, from insect feeding in the field. And anything that I saw naturally occurring was far less than 25%. So talking about my comfort level, I feel really comfortable with these levels that I simulated on the crop, because they were far greater than anything I saw naturally occurring. So again, the values were 25, 50, and 75% leaf removal. And the times throughout the season that I simulated this damage was 20, 40, and 60 days after planting. And so for the grain variety, that was 20, 40, and 60 days after direct seeding in the ground. And in CBD, it was 20, 40, and 60 days after transplanting cuttings into the ground.
And even
Thank
at
you.
75%, there was no difference.
No, it's, I hindsight here, I should have had a PowerPoint ready for you. And I could pull one up, but yeah, there's just no, I don't want to say no differences, there's slight variation, but I really think those variations in values are just coming from the plants being different themselves rather than the actual treatment that was applied to the plant.
Right.
Yeah.
That's wild.
It's awesome. Yeah. And I mean, I think it speaks to the resilience of the crop. I think it's a little more resilient than most, but having these data to go off of it's really supporting the fact that these can withstand a lot of leaf loss. And if it's beetles and grasshoppers feeding from your leaves and causing these big holes, or even kind of skeletonizing your leaves in the field, I really don't think it's a cause for concern based on these studies that I've done.
Yeah. What do you mean by skeletonizing? What does that mean?
So I basically mean just removing all the leaf area except for the vein of the leaf.
Gotcha.
Which
yeah.
You'll see that, and I've yet to see a plant that has had all of the leaf area removed off of it from an insect or whatever type of leaf loss might occur, So I think this study that I've done is a really, really extreme case in what you might see in the field. But
yeah.
Let's see. The question was, did you defoliate throughout the flowering phase? So with this study, the plants were only defoliated once. And it was either, 20, 40, or 60 days after planting. And at that value of either 25, 50, or 75%, only one treatment was applied to each plant. I didn't do it repetitively. But I will say, in reference to the flowering phase, in these timings that I chose, the 20-day and the 40-day were in vegetative phase, and only the 60-day was in flowering phase on both the grain and the CBD. So to be honest, in hindsight, if I could do it again, I would probably pick 30, 50, and 70 days. Because I think 50 is right about that time when they're switching from vegetative to reproductive. But hindsight is always 20/20, right? And I will say too, when we defoliated these plants, I defoliated every leaf on every plant within a given plot. So I think it was a pretty robust study design.
Yeah, it's interesting. So I know it's really common practice in cannabis growing, maybe not field grown hemp, but to really aggressively defoliate and pull branches off and take a ton of vegetative matter off. And I think that's, a holdout from the sort of black market, legacy knowledge, but it sounds like that's actually scientifically supported too. Like we're not hurting the plant by taking that much foliage off of it.
Just another anecdotal thought I have about this too, I'm wondering, I don't think that the plant should lose all of its leaves, of course, because photosynthesis has to happen in order for the plant to get nutrients to be able to grow and everything. But, you've all seen these plants growing, at the end of the season, they kind of naturally start losing a lot of those fan leaves. So, I think that supports the fact that this injury can occur prior to when, the plant just naturally loses its fan leaves and it's not going to hurt it. Even though, for some first-time growers or, people who are growers who are used to wanting a flawless crop, it's that this plant can really withstand a lot before the yield is impacted, and simulating this injury to this crop, it's like, I'm taking a lot of leaf area away here, even, drastically reducing the amount of leaf area that exists for photosynthesis to even occur. And it's still thriving. it's
Right.
pretty phenomenal.
Yeah, definitely. And that was some, very helpful insight from Suzanne, who had said something really similar, because I was really concerned at the end of harvest that I would have a ton of sort of dead or dying leaf matter on my plants. And she compared it more vegetable growing, where like, as long as the good shape, it doesn't matter. I know we're all obsessed with Instagram, but like, it doesn't have to look good for Instagram to be a good marketable product.
Right. Right.
And I was at an arborist meeting at some point over the last 10 years, and they were talking about, because they've done like looking at like gypsy moth, how they come in and, you want to talk about stripping trees, they leave them as sticks. And I can't remember, but they were saying like trees can withhandle having 100% of their leaves stripped off 1 to 2 times a growing season and not bat an eye. That, there's enough energy stored in these plants. They're getting enough nutrition from the soil. I mean, you don't want to obviously strip them 100%, but the plants are a lot more resilient than people think they are. And plants put out a ton more leaves than they need because they're designed to be part of the ecology and be fed on by insects. This is, what happens. Insects feed on the plant, the poop falls to the ground, it makes fertilizer. It's the whole circle of life kind of thing. But cannabis growers, and I say this to a lot of my growers, go smoke some of your own product and relax. It's okay not to have every leaf 100% perfect. And it is an amazing resilient plant because, I look at the levels of broad mite we see in cannabis, and if you have like a fraction of that in ornamental, the plant's completely destroyed. Cannabis is a really hardy plant. Anybody else?
That made me think of one more thing as we were talking, though. I'll just talk for just one more minute about why I chose this methodology. So, in the beginning, we thought about using a hole punch thing or something to remove leaf area, but we ended up using the shear so that I could, directly quantify the amount that I was removing from leaf to plant, just to keep it consistent. But we also thought about removing, pulling off the leaves completely, but I thought that might elicit a different type of plant response. So that's why we just use shears. And I know it's not, it's not actual insects feeding and removing the leaf area. And, when you cut these leaves, it doesn't look like it looks when an insect feeds as well, but that was done just so that I could quantify it from leaf to leaf. And like I said, I didn't remove complete leaves because I thought that might elicit a different response. So I can't speak to what might happen if you, just pull leaves off. But
that's a very common practice. That's more of what I think people think of defoliation, where they remove the petiole along with the leaf. So just to clarify, you're removing 75% of the leaf and leaving 25% in the petiole on there, right?
Yes. Yeah.
Okay.
So like if my fingers were the leaf, it's cutting off.
Gotcha.
Portion. Yeah.
Thank you.
Any other questions on the defoliation stuff? I see everybody taking notes, and thank you. That makes me, it makes me happy when I see people taking notes. So
makes me feel good. Yeah,
it does. But believe anytime you're a presenter and you see somebody write something down, it makes you feel very good inside because you feel like you've said something important enough that's worth writing down. Yeah. So,
just one question about that. I just wonder about actually taking off a portion of the leaf by cutting it, or having an insect actually damaging it by sucking the juices out of it and it's trying to repair itself. Is there a difference there, do you think?
Yeah, that's what I was saying is, I can't be 100% sure, but in the literature that I've surveyed, so I'm going to try like the crops that I can remember off the top of my head. I know these studies have been done in potatoes, corn, sugar beets, sunflowers. I know there are more, but that's all I can remember off the top of my head. But, they did this same type of injury where they just cut from the leaves, and I know I can speak about potatoes because I've worked with those, but, they've got beetle pests as well that have those same type of chewing mouth parts that are going to be putting potentially toxic saliva when they're chewing on the leaf, and by doing this, clipping, cutting damage in potatoes, they were, not at every point, but there were points throughout the season when they inflicted this damage that they did see negative results. So, I did the same type of methodology in hemp and did not see those negative results at any point throughout the season. So it's because of that comparison between the study methodology and the different crop types that makes me feel comfortable that even though I didn't use, insects to cause this injury, I think it's still a good indicator that the plant can tolerate it pretty well.
Yeah. Yeah. Well, over the years, I've definitely had some like serious mite damage, and, it would appear like we definitely lost yield due to that.
So that's a good point. When you're thinking about pests like mites or aphids, they have a different feeding behavior than these chewing insects like beetles and grasshoppers and caterpillars even. So they instead, they have a needle-like, I always just put my hand on my mouth, but a needle-like mouth part. It's like a straw. Instead of us, chewing with our mouths, like chewing on a hamburger or something, the way they feed is using pretty much a straw, and then they'll pierce into the stem or leaf or floral portion of the plant and pierce it with that straw and suck out the nutrients. So that's how they're feeding. Those studies are ongoing. I haven't conducted studies with that type of feeding behavior, but other people at other universities are starting to do that work. And it's a good question, if a spider mite or an aphid or any kind of mite feeds from your plant, it's not going to defoliate it like a beetle or a grasshopper or a caterpillar, but there's the potential that it could affect something that we're still actively learning more about.
Yeah, and with like aphids, even though I still do not think foxglove aphid is in cannabis, I'll have to agree to disagree with some people, because I think people that identified as foxglove, it wasn't foxglove, it was another species. But like foxglove aphid, though, in ornamentals, it has a pretty nasty toxin in its saliva, and when it feeds, it injects it into the plant, and it makes it very distorted, where generally from like cotton melon and green peach, you don't get that. So here you have different species of an insect feeding on a same crop, because you can have green peach aphid and foxglove aphid right on peppers. And if green peach feeds, you don't get distortion. If foxglove feeds, you get distortion. So the feeding damage can be different with these piercing, sucking insects and toxins in their saliva and what they're doing. And with the spider mite feeding, I mean, it's basically sucking out the contents of the cells. So it basically kind of stops the plant from photosynthesizing. And that's, you lose the chlorophyll, or are they chloroplasts, I guess, maybe technically, inside the cell. And that's why the feeding, it turns white, because you've lost that green content of the leaves. And how efficiently can they photosynthesize then? I'm sure there's been lots of studies done on other crops outside of cannabis, but it'll be interesting to see, how cannabis does. But also it's hard, sometimes you say, oh, we had spider mites and, our yields were off. Well, could it have been something else? And that's why doing it in a research environment where you have a control to see if something about the way you were growing or the genetics or something else can look and see if that really was what caused the problem, or was there some other outside force that wasn't taken into consideration at the time. So
that makes me think of, I don't want to open this can of worms. I know we're talking about certain things, but the chlorophyll or chloroplast point makes me think too, it's a good point to emphasize, because that's kind of what we see with russet mite. It's causing that gray coloration on leaves where it's fed, and it's making me wonder if it's like harming chloroplasts or chlorophyll production. But yeah, no, that's a conversation for another day. I'm not going to open that can tonight.
Well, does anybody have any other questions on defoliation stuff? And hopefully this was useful information. And I only bring the best people to my meetings. So anything? I hear your dog, Katie.
I know, I was about to mute myself. Sorry.
No, it's fine.
He just loves everybody.
Yeah. No, okay, so let's talk about some of your stink bug stuff, because I know indoor growers don't have as much of an issue, but it's also, again, very interesting because I do get, now I'm not getting them, but during summer, I was getting stink bug questions all day about, what is this? And it's hard when they're immature stink bugs, when they're in those first and second instars, trying to identify them is challenging. But there is a stink bug ID guide out there for free. Do you remember who put that out?
Yeah, my advisor is part of who put that out, my lab here. Yeah, I'll find a link for that, but I will make myself available. I feel pretty confident with stink bug ID, so I'll share my email address and all that. Feel free to reach out anytime. I love talking about hemp, and I love helping growers with insect problems in hemp. So anytime, I'll be happy to help ID those. But yeah.
Well, I'm going to be one step ahead of you because here's the link for the stink bug ID guide, because I have it listed on my website under my useful books and PDF link pages. So there's a link to that PDF you can download for stink bug ID. But so let's talk about what you guys were doing with stink bugs.
Yeah, so stink bugs, if any of you grow outdoors, I can't say with certainty, but I feel pretty confident saying that if you grow outdoors, you're going to see stink bugs or lygus bugs or some type of plant feeding insect in your hemp. So to try and figure out what's going on here, we did a study particularly with brown marmorated stink bug. So I'm not sure where everybody is from tonight, but brown marmorated stink bug is an invasive stink bug species. It's been in the United States for several years now, and it has absolutely wreaked havoc and caused all kinds of problems with a lot of commodities that are popularly grown, from fruit trees to vegetables to row crops, you name it. Brown marmorated stink bug probably feeds on it and probably negatively impacts it. So where I'm at in Virginia, I'm located in the mountains of Virginia at Virginia Tech, and we've got a lot of brown marmorated stink bug present here. So what we wanted to figure out, there were two key questions. One thing, we wanted to know if brown marmorated stink bug could conduct a life cycle, if it could live and survive exclusively from feeding on hemp. And then second, we wanted to see if we exclusively cage these stink bugs on seed heads of hemp in the field, are they going to affect that yield? Are they going to impact those seeds at all? So this hasn't been done in CBD, by the way, but we did it in grain, and I feel like that's still pretty similar. So anyways, the first part of the study, we reared brown marmorated stink bugs exclusively on hemp or exclusively on corn, which is a known host plant. And what we were able to gain from that study is that not only were they able to survive on hemp, but more of them survived exclusively on hemp than on corn. So that lets me know that hemp is a suitable plant for growth and reproduction for this insect. And then the second part was, like I was saying, we caged brown marmorated stink bugs on seed heads of grain hemp in the field. And we caged them in densities of either, nothing for a control, 10 brown marmorated stink bugs, or 20 brown marmorated stink bugs. And at the end of that study, we did see variations in the data. But like I was saying earlier, with the defoliation study, I really feel confident that those differences in yield were just due to variations in plant density in general, not due to the insect feeding on the plant. So that was just in grain, but at this time, I really don't think that stink bugs are a concern in hemp. And I can say anecdotally too, you walk through any kind of outdoor growing situation and you're going to see an abundance of stink bugs or lygus bugs. And, these are somewhat puzzling to me just because of the effect that they have on other agricultural crops. They seem to have some type of negative impact on whatever crop it is, but I've yet to see any negative impact of stink bugs, of lygus bugs, of any kind of plant feeding insect in buds of hemp, whether it's grain seed heads or it's CBD buds. And, to throw a disclaimer, this could change in the coming years, as hemp becomes a more regularly rotated crop within United States agriculture and Canada agriculture. But right now, I'm not able to see anything negative from their presence or feeding in the crop. I hope that's helpful. I will also post or share a link in the chat of the study that we did with stink bugs.
Yes, and if anybody wants a cup of stink bugs, you just come to my house and I will fill it up for you. Because we are 2 miles, I think, from where the first brown marmorated stink bug was found.
Allentown.
Yeah, well, it was actually out here near Gurmansville. It's like, because no one's ever heard of Gurmansville or Slatington or these little areas out here. Yeah, and when Penn State said, oh, it's not a problem, don't worry about it. And here we are, how many billions of dollars later? Yeah. They're in all my shoes every fall. They fly around at night when you're trying to read on your tablet. Yeah, loads of fun.
Yeah.
So, because I know everybody's head I see on the screen. I know you guys are indoor growers, but again, I've had some guys contact me about stink bugs getting in, and then they freak out because there's one stink bug in there. I'm like, just catch it, put it back outside. Don't worry about it. And it's not anything that you need to be so worried about it. But I do know some of them do grow outside too. It's just the people that have their cameras on right now are more indoor growers. So, but again, even if you don't have stink bugs now, it's good information to file away. So if it does become an issue, but I do think ligus is one that I've seen in insane numbers indoors in cannabis facilities. And the one facility we collected samples from, and it actually had a Western tarnished plant bug, and I can't remember the other species, but 2 different. And the immatures can kind of look like aphids. So, and they have the same kind of piercing, sucking mouth parts, but they run a lot faster than aphids. Aphids are pretty slow. The ligus bugs, to catch them, I was out there with my aspirator and, making yourself about pass out because you're just sucking, trying to get these guys sucked up into the container. But the grower was convinced that from over time, the feeding damage would actually cause some of the plant tissue to kind of, I don't want to say go necrotic, but close to it, and kind of fall out of the leaf and kind of give it kind of like a skeletonizing look a little bit. But that wasn't 100% confirmed for sure, but they had that damage and they had heavy ligus, because it would be like if somebody went along with a hole punch constantly, because I would say on, a leaf frond, there was probably 5 or 6 ligus per leaf. I mean, those kinds of levels.
Never say never,
but they also had broad mite damage. So they had multiple issues. So it's, maybe it's the combination of everything, and maybe there was nutritional problems. It's hard to know.
Right.
But with the ligus, and this is the problem for indoor and even, marmorated stink bugs and green stink bugs, there's nothing you can really use to treat for them indoors, even outdoors. I mean, what are you going to even do about it?
Yeah, that's a good point. And I'm going to throw another link in the chat. I've done a lot of insecticide efficacy work within hemp to manage specific pests, and I have published those, and they're open access. So I'll give you a link to the journal, and if you just search for my name in the journal, it'll pull up the ones that I've published from my lab here. But so back to the stink bug point, for the record, it's really difficult to manage those in crops, because the options that we have of what we can even use in hemp, they aren't really cutting it with managing that pest. So a little bit about insecticide work. We'll do efficacy trials in the field where we apply these products and then count numbers of insects from plot to plot and see how well these products are helping to manage some of those populations in the field. But another element to insecticide work is that you can do laboratory bioassays. So that is a super controlled environment where you're treating hemp with these particular products, and then you're feeding that treated hemp to the target insect that you're trying to see whether or not the insecticide will help manage it. So it's best case scenario with these insecticides when you do laboratory bioassays. But with the stink bug bioassay study that we did, we only ever saw 60% efficacy at best at helping manage these insects. And that's good, within a crop system. So I guess what I'm trying to say here is that I don't really think they're worth managing when you see them come along. And that's a good thing, because the products that we do have that are labeled to manage them, you're not going to get the efficacy that you're desiring from those products that you have to choose from.
Well, for my indoor guys, I've had them vacuum them off.
That's a much better plan.
You can go to Home Depot and get those tiny little shop vacs, and you guys are smart, put some kind of throttle so it's not going to suck the leaves off your plants, and find that where it's enough to suck the insects off without sucking your leaves off. And that can be effective for removing ligas. But I would be interested down the road to see. So, we always talk about a lot of Bavaria bassiana work, and how we've been talking about how we can have biotypes of insects. You have cultivars of cannabis. There's strains of Bavaria bassiana, and the one that's in Botanigard, that's the GHA. The strain that is now in the Bioceres product was actually isolated in Quebec out of a tarnished plant bug.
Wow.
And so I'd be really interesting to see a trial looking at the Bavaria bassiana strains. Now, it doesn't always work out like you think it's going to work out, because Isaria, the fungus that's in PFR 97, was isolated out of mealybugs in Florida, because Dr. Lance Osborne kept losing his mealybug colonies because this fungus was killing it. They isolated at PFR 97. It turned out to be PFR 97. And that's the Apopka strain.
Yeah.
And we're like, oh my God, yes, we're going to have a product for mealybugs. It doesn't work for shit for mealybugs out in the real growing world, unfortunately. So it doesn't always work that way. But I don't think there's been enough work yet looking at the different strains of Bavarias to see if there would be a difference. But the fact that this was isolated, I actually know the person that isolated up there in Quebec. And it'd be interesting to see how it does perform under right environmental conditions, to see if it could knock back the tarnished plant bugs in a growing situation. No guarantee because of what happened like with the mealybugs, but it would be interesting to look at.
Yeah. I'll make a quick comment on that. So I did a larger scale field insecticide trial this summer to manage corn earworm in outdoor grown hemp, which is just a problem pretty much everywhere. But the reason we put a Bavaria product in the trial, and the reason was to see if it helped, if it would show any activity toward stink bugs or tarnished plant bugs in the field. So for what it's worth, just here where I conducted the trial, there were never enough of those species in the trial to be impacted by the Bavaria application. So hopefully that'll calm your nerves a little bit. But additionally, if you're growing hemp outdoors and corn earworm is an issue for you, I know that the Bavaria products can be a little more pricey, but it was pretty exciting and incredible. What we saw is that when the Bavaria product was combined with another product where the active ingredient is a virus that exclusively, did I freeze up?
You froze up, but we can still hear you.
Oh no.
We can hear you.
Yeah, I was like, oh crap, everybody else froze up. Okay, so we mixed the Bavaria product with the virus product that impacts corn earworm, and we're seeing really good efficacy out of it. But when that virus product was combined with the Bavaria product, it worked really, really, really well. We're not sure. I've been talking with another researcher here who has a history of working with insect pathogens like Bavaria bassiana. So we're curious and want to do further studies to see if there's some type of synergistic effect when those two are combined. But I guess the main points out of that were to tell you that we never had super high infestations in the field. But for what it's worth, when it's combined with the corn earworm product, it works extra well.
Well, it seems Bavaria does that. I mean, when you combine it with an azadirachtin, that ups its game much better too.
Yeah.
And I don't know if you've been by Vestron.
It's Spearlip, Vesteron,
Which
Spearlip, and Spear T.
Spear T, because when initially, when we're like, why are you even presenting this data? It was useless. But where it was interesting is when they did combine it with Bacillus thuringiensis, the bacterial toxin for caterpillars, where there was resistance, it kind of brought the product back to life. So I do think that these, more so than without it. But I mean, you haven't seen that product take off in the market at all. And it's been around for 6-7 years now. But I think there are a lot of synergisms with these products that have yet to be explored.
Yeah.
Especially with the microbial products. I think there's definitely something there to it, because I mean, hey, it's like if you're down with COVID, maybe you can get over it. But if you have COVID and flu, you're done.
Yeah. Yeah. Side note, COVID is a really helpful tool in helping explain some of these active ingredients these days and how they infect insects. Yeah, it's a benefit.
So cool. No, so I think that was useful information on that. And again, there's still going to be a lot more research coming on all this stuff over time.
Started. Yeah.
And we're just getting started. Like I said, I've had some pretty good calls over the last couple of weeks with private industry, bug people, and also some university stuff about directions to go. And one of the more shocking things was the lack. So, well, there's the IR4 group, a project. I don't know, call it project or group or IR4 program.
IR4 project. Yeah.
Which basically helps specialty crops get labels, because companies, the big agricultural companies that make pesticides for corn and soybeans, they'll pay to register a product there because they're going to make a ton of money because they got to treat bajillions of acres. Are they going to get paid to get that product registered for geraniums? No, because they're never going to sell enough product to recoup the registration. And they were just open for comment for the IR4 about hemp and getting products to test what products for what pests. And this is what somebody and I were emailing about, the fact that this huge lack of products in the IR4 for this year for hemp that people weren't putting in, saying we need these products to be tested on hemp pests, which is a little scary, because here we have the opportunity to have the research paid for and done. Like, does azadirachtin and Bavaria bassiana work on cannabis aphid to get it a hemp label for it, but no one is putting in for that work for it to be done.
Well, to make a comment, we did do IR4 work this summer, and I thought there would be more from it. The focus of IR4 this year was corn earworm, which is it's a huge issue in hemp, but yeah, there's more than just outdoor grown hemp. There's more than just corn earworm. There's russet mite, there's cannabis aphid, there's lots. So just to comment and say, it needs to expand. And
I have to check because I think the closing date's coming up for input for next year. And and this is where the cannabis industry needs to come to these groups and say, here's the stuff we need, the way that poinsettia growers are organized, the way, floriculture, the tomato people, those guys all come together, because there isn't enough organization yet, I think, in the cannabis industry, just because it's still, in its infancy.
The baby, yeah.
Yeah. So, but does anybody have any other questions on that particular topic? Because I think we'll hit one other thing, then we'll call it a night, because, I don't want to keep Katie too long. I'm sure her dog misses her. And my Coke glass is almost empty, which is a crisis. So does anybody have any other questions on that? I saw Andrew's camera come on. Does that mean Andrew has a question? I see his eye in the corner of the screen. He's coming. Okay. Did you have a question? Or are you just coming? Okay. Well, I just thought maybe you're coming back. She had a question. Yeah, we can see you. Remember, we can see you. All right. Well, then last thing, I just want to really quick touch on our beloved hemp russet mite. Just because you did some trials on looking at spray products, and I know that I know some of the products you did in your trial is nothing that's ever going to be registered, so you do not have to speak to those because don't give them any illegal advice on that. But I think it's kind of important to mention that Suff Oil X was in the trial for hemp russet mite, and how'd that do for you?
I'm going to paste in a link, the russet mite trial, but there were a couple that stood out. So yeah, if you follow that link, you'll see the article, and at the bottom there's a table, and the one you want to pay particular attention to is the December 3rd, which is 22 days after the treatment was applied. So to set up this trial, what we did, or what I did, I applied these products twice. Oh, did I? No. Okay. I applied these products twice, 2 days apart, and then we 3 times after the fact. So when you're looking at that December 3rd date, the numbers that have a D next to them, which are PLP, natural, mammoth, you've all probably heard or seen mammoth biocontrol, Agrimec. Don't use Agrimec because that's a conventional product. Movento, and then Suff Oil X. Those all resulted in the fewest number of russet mites 3 weeks after the treatments were applied. So take it for what it's worth. Of the products that are actually allowed, I think the only things that are technically allowed due to their label language, Suzanne, you can back me up on this, but I think it's going to be Grandivo and Venerate.
Well, and it's going to depend by state and who you are. So that's for hemp. You're speaking to hemp. I'll speak to the cannabis stuff because, again, it kind of convinced through the states, but you can use, sulfur, impede, mammoth, Suff Oil X and things like that. Grandivo and Venerate in some states out there. So, hey, did you and Whitney get together and compare your trial results on this compared to what he did?
No, we haven't had that conversation. I know he did the trial, but it's not public.
They didn't publish the information. I mean, he came out and said that sulfur and Suff Oil X were his 2 best in his trial, were the results, which obviously you cannot use those together. So you have to commit to one direction or the other on those. And I get really nervous about sulfur. And one of the discussions we've talked about is there's really not a sulfur label for indoor use for this application. So there's a lot of discussions about that too. But just be aware that there are trials going on with this stuff. And I'm sure there'll be more trials over time. I still, at this point, feel there's no biocontrol agent that controls hemp russet mite that is commercially available.
Right. Which is the catch, and it's the important catch. Yeah. Right.
Because as time goes on, we're going to find things out in the field eating it. And I know there's been one mite collected, but they've not been able to identify it.
Yeah. Another thing, I'll try to find this before we end today, but this is one in particular, like I was talking about earlier with cannabis aphid, we've got to understand the pest and its life cycle and its biology in order to be able to understand how to manage it efficiently and effectively. But we know even less about hemp russet mite than we do cannabis aphid, which is, it's just the way it is because of prohibition and all of that. But, we're working on it. But, the one paper that exists about hemp russet mite, there's a line in the paper about how it can be managed with, is it CO2, I think? And I don't know exactly how you go about that, but for what it's worth, I've never done that. I don't know how it's done, but I'm going to try to find that paper and share it.
Well, and what would be, see, the thing that I always think about the eggs, because, you know, if killing the eggs is so critical, and you think of the respiration rate of eggs, and it's so low. And this is something Whitney and I've talked about quite a bit. And that's when he was seeing the differences between the oils, because he even said that, you know, sometimes you need 2 applications of a product over the egg to suffocate, to get enough on it to suffocate the egg enough, because the respiration is so low. And I think about that with like CO2 treatments, that maybe you can knock out the adults, but you're going to have to come back and do it again, and probably again, to be able to break that life cycle completely. And
it's just tough too, because, you know, the limited experience I have with it, like speaking from, I mean, you could probably back me up even more, but, you know, I understand sometimes like you have to reuse soil, you know, due to economic costs and all that, you know, there are just issues that exist. But the grower that I was able to work with one-on-one, you know, they didn't have any presence of this mite at all. And I think what kept it coming back was that they kept reusing soil. You know, they would eradicate it, but then it would be present in the soil, so it would continue to exist and it would continue to be a problem. So it's, you know, you want to do everything that you can to prevent getting it in the first place, whether that's quarantining plants, inspecting them, making sure you've got a microscope or some kind of magnification to be able to see it on transplants, or, you know, whatever you're bringing in, because it's, you know, especially with indoors, which seems like the majority of y'all are indoor growers, but once it's there, it's so hard to get rid of. And, you know, I don't think it's a complete death sentence for plants based on what I've seen, but it will really knock back the yield.
Well, but I think you have to have a pretty good population. But the problem is growers don't realize it until they're in flower.
Yeah.
And then that's when I get the call. I'm in flower, and what's going on? And because they're not being proactive enough on the earlier stages. And just to clarify, hemp russet mite is not living in the soil, but I think when you harvest plants, they can fall on the soil. And then all it takes is, you know, a couple getting back up on the plant. Because I've had people, well, it will affect, Shane, it will affect your yield and quality, but you have to have a certain number of them before it happens. And if you look up on one leaflet and you see like 6 or 7 hemp russet mites per leaflet, that didn't start last week.
Right.
You've had that for a long period of time, and it builds up and builds up. And once you tip that economic threshold is when it becomes a problem. And the problem is you have to stop it before it comes to an economic threshold, but people with the hemp russet mite don't realize they have it till then. This is the problem with it, because it's so tiny, because it's really hard to scout for and really hard to detect because of its size. I mean, the same thing with broad mite. You don't know you have it till you start seeing the damage. And that's why floriculture crop people now like know the crops that get broad mite. And so they do preventative treatments absolutely before it even starts. Because I mean, I've seen bud come to harvest like that big that had damage. So, I mean, it absolutely will. But again, it's a numbers game. And sometimes when people contact me and they're already in flower, it's just band-aiding to get to harvest, because you're not going to eradicate them because you're so limited on what you can do. But if you can get your lowers low enough that you don't have an economic impact and you still can get a good harvest, that's kind of what we're doing. But then get that complete cleanup. But it's challenging when you keep soil, because how are you going to guarantee they're killed on your soil surface? And someone did mention about green cleaner that can be applied every few days. Well, it's a soybean oil. And I mean, that's the thing with oils. They're a good suffocant, but you shouldn't have to keep applying it. And I worry sometimes with some of the soybean oils because they're heavier and they're not as emulsified as well. And there are concerns with phyto, which maybe you won't get in an indoor setting, but if you're in a shade house or something, you do have to be careful with. But the oils, I think, are the better way to go for hemp russet mite at this point. And it allows you to continue on your bio programs for your other pests, where if you use some of these harsher products, those will completely derail your other biocontrol programs. So it's a tough balance there, because if you get hemp russet mite, you have to use some kind of spray product. Whether I've had one person, they used repeated cold water sprays to get their numbers down enough so that they could get to harvest, and it worked. So not just necessarily spraying a pesticide, but things like that to get it down. Someone said they've had a hard time getting full leaf coverage with Suffoil X. Maybe you need to defoliate so you can get into the canopy. And also, again, this is where spray equipment is so important. The Zoom we did with Kurt Becker, I don't know, like 2 months ago, was so phenomenal about particle sizes and PSI and all that kind of stuff. Because again, according to the research, and when these researchers are testing these products, they're getting coverage of these pests. And that's what, they're testing to see if the products can kill these things. And they do. But then it's really hard then to get that 100% spray coverage out in the field. And that's why with younger plants, there's so much interest in dipping, because that does give you that 100% coverage out there. I fear doesn't get a good spread. Okay, I was saying, I don't understand what is, whoever,
He said not fear.
The fear spreads pretty good depending on the pest.
The fear spreads. I still don't understand. I'm sorry. The fear doesn't get.
It's more of an existential.
Okay, I'm going to go with that and just let it. Okay.
Suzanne, is there any way to do early detection on like russets or broad mites, or is it?
Yes. You don't want to do it.
Sure, because they're so tiny.
Yeah. You can do what is called destructive sampling and go take a bunch of cuttings off your plants, put them in alcohol, put them through this like chamber that has the right size of filter paper in it where the alcohol drips through, and then take each of these discs that come from each different, because you don't want to put all your samples in one basket, because then you don't know where the problem is. And then you have to take those filter discs and look at them under a scope to see if you can see the hemp russet mites on there for early detection.
Gotcha.
Sound like fun? No. I would have good sanitation practices and have a quarantine area. I cannot say this enough. And I just had somebody call me, and they put their quarantine area in. They took in plants from somebody. They had them in the quarantine area and found out the grower had hemp russet mites. And they had not put them in that quarantine area. They could have given, I'm sorry, not hemp russet mites, sorry, root aphids. They could have given them to their whole facility. And they're a living soil people. And once you get them in living soil, so I cannot say this enough. If someone's going to build you a greenhouse and they don't have a quarantine area in it, or a grow without a quarantine area, you need to find another designer engineer, because that should be standard in all facilities, is a quarantine area.
Yeah.
What does the quarantine area entail? Because like I started working at this facility, it had already been built and operating in its capacity. So if we were going to retrofit and put one in, what would it look like? Not that I'm bringing in plant tissue, but
it's just good to know. You just need an area is a small individual grow room that has, grow lights, irrigation, your typical setup, but that if you can leave them there, and then if there's a pest problem over a 2-week period, it will come to fruition. And that way, you can inspect those plants thoroughly before they go out to the main population, or you can douse them with biocontrol agents or spray, whatever program you're going to do, just to make sure you don't introduce them to the main area. It's like when you go to a foreign country, rabies, they hold animals for X amount of time. You want to go to Australia right now, you go from the airport right to a hotel room for 2 weeks. You don't even get a room key. That is your quarantine area to stop the spread of COVID. Back to Katie's point, we can relate to COVID for so many things these days.
Yeah. Same concept.
Because the expense of doing that is going to save you in the long run. In regular floriculture industry, when plant material is moved around, you're required to have what are called phytosanitary certificates. State inspectors inspect your plant material. Any of the offshore cuttings that come in get checked by the USDA to check for pests. None of that's going on in the cannabis industry. And that's why these pests are so freely flowing, because if let's say you're in Florida and you want to send a whole bunch of plants to Canada, you betcha those plants are going to be inspected. And if there's a pest problem, either they'll get destroyed at the border or sent back. And again, nobody's doing this kind of stuff in cannabis. And this is why it is like the freaking Wild West. And the problems are getting spread from one end of the country to another. And people are not following these stuff. I mean, there needs to be phytosanitaries for movement of the cannabis plants, but cannabis isn't federally legal, so they don't fall under this, phytosanitary program.
Right. Yeah. Well, a friend of mine, they were helping set up operation in Oklahoma, and their consultancy that was helping them set up the grow is based out of Oregon, and they just drove, a moving truck full of not just like mother material to start with, but like, here's your entire first crop's worth of clones, all the way across the, the country. And, that's all I thought of when they told me about that. I was like, well, we start from seed.
Yeah. I can't even. And especially, not that I don't love you guys in Oregon and Washington and the West Coast, but on a scale of 1 to like 10 buggy, it's like 15 out there. Yeah, that's scary. But I will say that once the quarantining with COVID and all this, and I could start moving again, I do think Oklahoma is probably in the most need of me at this point. I need to go out there and teach some classes. I've decided I'm probably just going to go to like Holiday Inn, rent a conference room, and, registration fee, I'll figure it out, and then just come spend a day with me, because just the baseline knowledge. And I'm going to try to do this in a bunch of states to get everybody up to speed. And I do accept gifts of like soda. In Canada, I'll take butter tarts. You can bribe me completely with that kind of stuff. I do accept bribes to come.
Good to know.
So, yeah.
And Wonder Bars.
Yes.
Ooh.
Wonder Bars are on their way. December 14th. It said delivery.
Yes. And
you'll be getting a double load. There'll be some there on the 10th.
Yes. I'm going to put my fat pants on, which have cats and Christmas hats, and I'm going to lay on my sofa. I'm just going to make a video of me shoving Wonder Bars in my mouth for y'all. Oh, yes, horseback riding is also an excellent bribery tool for me. That has worked in Montana, but I will go horseback riding anywhere anybody wants. I used to teach horseback riding, so I miss having a horse, but I travel too much to have horses now.
Well, I'm inviting myself on this next trip of yours.
Yes, my one bucket list of horseback riding. I got Montana. I've done Hawaii. I want to go ride, and I've never wanted to go to Jamaica other than the reason to ride a horse on the beach in Jamaica. That's the only reason I want to go to Jamaica. And Katie, you want to go with me? We can go together down there and do that.
We deserve it once this passes.
And we can go look for bugs too.
Perfect. Yeah.
So cool. Well, does anybody have any other questions? Because me, I'll talk for days. But we've been on for 2 hours, and I don't want to our numbers are slowly dropping. We're down to 25 people. No, you guys are the hardcore people. And Matthew registering at the last second tonight. I saw that. And also, Chaz, you were pretty late in registering too. Don't wait to the end. Yes. I always have to chase Matthew down to make sure he knows to register.
Hey, I registered this morning.
I didn't get the thing till this evening.
No, I checked in early, at 5:30 or at 4:30 or so, but I did it this morning when I woke up just to appease you.
Thank you. So, all right. Does anybody have any other burning questions for Katie or myself? Make sure you like my candy Instagram page so I can get some more likes to legitimize my candy making as a hobby.
Thank you, Katie.
Yeah. I'm about to give Katie the huge thank you for coming to do this. We've been talking about this for quite a while, and she and I have spoke at some meetings together, and it's been awesome. But, I bow at the temple of Katie and for the work she's doing, because it's so essential, and we need people like her to be doing this very targeted research. So thank you.
Thank you. Thanks for being here. Yeah, I know it's been 2 hours and it's late, depending on where you are. There's still so much that I don't know. That's the thing. Like, you can study this crop and the pest problems for years and years and years, and we're still not going to get to the bottom of it all. But this crop means a lot to me, and being able to speak with you all one-on-one, it's special to me, because I know, there's a lot of unknowns, and I want to give you all the information that I've been able to get so far. And, we've barely scratched the surface on even figuring out what questions need to be asked. There's just so much work to be done. But yeah, I've had a great time with you all. It's been a good conversation, so I hope it's been helpful.
Yeah, and when you have more stuff you want to talk about, just bring, give me a call and we can, have it come back and do this again. Because with the university and extension people, because it's so hard for them to get funding, and they live or die by funding. And as we as people don't let grant money know where it needs to go and support and turn out and support the researchers, then they lose the funding. Just like in Rutgers, New Jersey, they got rid of extension because they didn't feel it was needed anymore. And it's so needed. And I don't think these researchers, there's a disconnect between the industry letting the right people know that these people need funding, because a few of my friends that are at universities say they spend 50% of their time writing grants to get funding to do research. That means half their time they're out there trying to get money instead of getting more research done. And there's so few people doing this research we need. So Katie, if there's ever anything I can do to help support you, and I can, help, network with the cannabis industry, you let us know, because we will be there for you. If you ever need us to write requests, if you need us to help fundraise, anything like that, we'll be there to support you.
Yeah, I'm glad to backtrack just for a quick second. I'm glad you brought that up, because I did want to say, I've been really lucky that I've been able to do this work as a grad student, because, in order to support a grad student, a professor has to have a considerable amount for this type of research, where I can tell y'all too, I'm still a student, but I'm a PhD student, so I can carry a little bit heavier of a load. But I've, contributed the text to every hemp grant that my advisor and I have submitted. And the amount of funding that we've gotten compared to what we've applied for, we've gotten like 2 grants out of 10 or so. And that's no exaggeration. It's so hard to get money for this. And we realize how important this work is, and y'all realize how important it is, but trying to sell this to the USDA or even a state level granting agency, it's just somewhat impossible sometimes. Like, we can argue the point for just pages and pages and points, and they don't always get it, because, for better or for worse, whatever, this crop is still stigmatized, and they just don't see the need. And, we've tried to sell it in every guilt word, every word you can put in a grant, and it's just really hard to get money for. So just letting y'all know that, even the research information is slow coming out of our institutions, but, we're doing what we can. And yeah, I've really fallen in love with the extension aspect of the university, and it's what I want to stick with. But we've got to, find the funding to keep my ability to do that within a university realm. So trying to get out everything I can.
And it's expensive too. And I don't think people always realize it's not just the salaries of the people. Because I was discussing with another university researcher about, again, doing some root aphid research, and you need a technician who's the person that's the I say grunt work, but that's the person that waters the plants and does counts and all that kind of stuff. And I said, if we wanted to do something like that, how much money would we need? And because the university takes a chunk of the money that you actually get, they would need to bring in like $70,000 or $80,000 just to get a technician to be able to do that. And the technicians, making probably minimum wage, or just a bit above that. It's not a high paying job. And so it takes a lot of money just to get a little research done. I wanted you guys to be aware of that, because, we need to find ways to help support this industry, because these are the universities are where we need to get research done. And, maybe I just need to win the lottery and then we can have a Bug Lady grant and I won't make you do paperwork.
Yeah. It's just gift money.
I wish I could gift money. But that's a discussion we've been having with some of the other researchers, is if I do fundraisers through the cannabis industry and then I gift it, because if you gift the money, the university takes less of it. And it's a way to get more work done. So there's some discussions on that. We've been talking about this like for 2 years, but it's a lot of paperwork on the university end on getting things in order. So there may come a time where I may put out a begathon to say, we're going to raise money for Rudy for research, and just everybody kick in a little money. Because if all the cannabis growers kick in, we could get enough money to get some good research done. And that's what I'm trying to make happen. So, well,
Okay.
All right.
Yeah.
Well, thanks again, everybody. And thank you, Katie. I really appreciate it. You have a good night, and you haven't sent me any new photos of your dog lately. You did not share your dog photo.
I will, this is my dog, Scarlet.
Those ears are just,
The border collie. She's just over 6 months. We got her, but totally because of COVID. But oh my gosh, I've always been I always loved dogs, but I've been more of a cat person. But now having her, I'm like, I'm definitely a dog person. She's letting, I'm definitely an extrovert. So now I go to the dog park all the time and like have all these new dog parent friends. But she's great. She's been the best part of COVID.
oh, cool.
Nice.
Y'all would love her.
I don't know if my cats would. My dog might, but I don't know if my cats would.
Yeah, we're still working on the cat-dog relationship, but it's a work in progress.
Cool. All right. Is it one last chance? Any burning questions or anything else from anybody?
Real quick, if I was going to send samples of the aphid to this Blacksburg address, is there anything that I need to include in the sample? Any information?
Yeah, I can type it into the chat, but if you could put, the crop, whether it came from hemp or marijuana, whether it came from indoor or outdoor, the date you collected it, the variety. Let me go ahead and type that in.
Yeah.
County.
Yeah, like GPS coordinates would be great, but anything close, like city, county.
Because not everybody's want to get you right to their doorstep, but they'll probably get you the county area.
Yeah. We have to have the secret knock, so.
Let's see.
Awesome.
Date collected.
And you can just use alcohol, rubbing alcohol. You can actually buy rubbing alcohol again. I've seen it in the stores. Well, there was for all us insect collectors, there was a huge shortage at the beginning of COVID.
Right.
So
they use 70%. We have a ton of 99, but I know you were telling me that kind of obliterates the bug.
Yeah, I think that's too high. And also, if there's a crack in the thing, it evaporates really quick. But here, let me show you. Let me show you not show you. I'll get you a link. These are what orders. These are the ones I've been ordering off. That's crazy. I order the vials off of Amazon. But I actually ordered the lids off of BioQuip because I found it was cheaper to do that. But for what you guys are doing, just to buy these and for quick shipping, I would just buy a case of these or a box of these. It's $29. And these little 2-gram vials are great for collecting bugs. And I don't know if you guys remember the technique, but you get the aphids and you take a paintbrush, dip the paintbrush in alcohol, then pick up the aphid and then dip it in the vial of alcohol, then pick them up because you don't want to crush the aphids. You don't want to damage the aphids. And using that little paintbrush pickup technique, which I have that buried way back in my Instagram account on bug collecting. So.
Awesome. Thank you, Suzanne. Thank you, Katie. This was super helpful.
Yeah. Well, good. I'm glad you guys got some out of it, because again, we just wanted it to be a little free flowing tonight and not do. I didn't want to make Katie have to do a whole bunch of work of putting a presentation together.
Well, I kind of wish I would have now, because it would have like supplemented my defoliation thing, but it's okay. I think. They
got the ideas, and sometimes people get tired of just looking at data. So no, I think it went great. So, all right. Well, thanks again for everybody. And you guys have a good week, because it is Monday. Yes.
Yeah.
Another one. So, all right.
Thank you guys. Good
You're
night.
dismissed. Thanks. Except for Katie. I want to stay on for a second.
Yeah, I was going to ask you to stay.
And Sam was really quiet. I don't know if Sam was really here or not. See, because I think sometimes people sign on. Oh, you